Hello, everyone. Welcome you to this presentation on a crucial component of regulatory pharmaceutical science, the ICH-Q3A R2 guideline. Today we will go through an in-depth walkthrough of this harmonized tripartite guideline focused on impurities in new drug substances. This guideline is pivotal in ensuring the consistency, quality, and safety of pharmaceuticals across major regulatory regions, including the European Union, Japan, and the United States. Let's start with the background. The ICH, or International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, is an international consortium that brings together regulatory authorities and pharmaceutical industry stakeholders. Its mission is to promote public health through international harmonization of technical requirements. Q3A R2 specifically addresses the control of impurities in new drug substances obtained through chemical synthesis. The aim is to ensure that such substances meet stringent quality standards before entering the market. It's important to emphasize that this guideline does not apply to biologically derived substances, peptides, oligonucleotides, radiopharmaceuticals, fermentation products, herbal products, or crude natural extracts. The development of Q3A has been progressive. Initially codified as Q3 in 1994, it was revised and renamed Q3A in 1995. This marked its recognition as a harmonized standard. The first major revision, Q3A-R, occurred in 1999 and was subsequently adopted in 2002 as Q3A-R1. In 2006, a specific revision was made directly to Attachment 2, resulting in the current Q3A-R2 version. This revision did not undergo public consultation, emphasizing the consensus already achieved among regulatory authorities. Now, let's clarify what this guideline covers. Q3A R2 is applicable to new drug substances that are chemically synthesized and have not yet been registered in any ICH region. It does not extend to clinical development substances or naturally derived products. The guideline considers impurities from two principal aspects. Chemistry. This includes classification, identification, analytical techniques, and specifications. Safety. This addresses qualification or the assessment of the biological safety of an impurity. The goal is to ensure that all impurities are detected, reported, and controlled to maintain product quality and patient safety. Impurities in drug substances are categorized into three broad classes. Organic impurities. These arise from the synthesis or storage process and include starting materials, intermediates, byproducts, degradation products, and residual reagents. Inorganic impurities. These include residual metals, salts, filter aids, and catalysts. These are usually known and can be controlled through routine analytical testing. Residual solvents. These are solvents used in synthesis and must be controlled in accordance with ICH-Q3C due to their potential toxicity. Excluded from this classification are polymorphic forms, enantiomeric impurities, and extraneous contaminants, which are better managed under GMP. The rationale for impurity reporting and control is grounded in scientific assessment and risk management. Applicants must provide a list of actual and potential impurities likely to arise from a synthetic process, supporting data from batch analyses, including those from development and proposed commercial processes, stress testing data to identify degradation pathways. The impurity profile must be compared across batches and thoroughly discussed, especially if any impurity exceeds the identification threshold specified in Attachment 1. In cases where identification is not feasible, a documented explanation must be provided. Analytical procedures used to detect impurities must be validated per ICH Q2A and Q2B. These methods should demonstrate precision, specificity, sensitivity, and accuracy. Quantification methods may include chromatographic techniques like HPLC or GC, spectrophotometric methods, Comparisons against standards using response factors. If the impurity and the drug substance have dissimilar detector responses, correction factors must be applied. Lower precision methods like TLC may be acceptable if scientifically justified. All analytical assumptions must be declared and supported in the application. Every batch used in clinical, stability, and commercial development must have its impurities reported numerically. The guideline advises use of 2 decimal places for levels less than 1%. Use of 1 decimal place for levels greater than or equal to 1%. Reporting all impurities exceeding the reporting threshold. In addition, data should include batch identity, size, and manufacturing site, impurity profiles and their origins, analytical methods used. Chromatograms of representative batches must be maintained and provided upon request. Specifications are the regulatory standards that define the quality of a drug substance. For impurities, the specifications should list each specified identified impurity, each specified unidentified impurity, any unspecified impurity with a threshold limit, total impurities. The rationale for each impurity's inclusion or exclusion should be explained based on safety, stability, and manufacturing experience. Specified impurities should be clearly labeled, and analytical detection limits must match the impurity's toxicity profile. Qualification refers to the demonstration that an impurity is biologically safe. An impurity is considered qualified if it was present in batches used in preclinical or clinical studies, is a known metabolite observed in humans or animals, has safety data supporting its presence. If an impurity exceeds the qualification threshold and lacks safety data, toxicological studies must be conducted. This includes genotoxicity assays and subchronic toxicity studies based on the patient population, dosage, and route of administration. Some important terms you need to be familiar with include impurity, any component not defined as the drug substance. Specified impurity, individually listed impurity with a specific acceptance criterion. Unspecified impurity, controlled by a general limit but not specifically listed. Thresholds, levels above which impurities must be identified, reported, or qualified. Impurity profile, a description of both identified and unidentified impurities present. This table categorizes threshold values based on the maximum daily dose of the drug substance. For less than equal to 2 grams per day. Reporting threshold, 0.05%. Identification threshold, 0.1% or 1 milligrams per day, whichever is lower. Qualification threshold, 0.15% or 1 milligrams per day. For greater than 2 grams per day. Reporting threshold, 0.03%. Identification and qualification thresholds, 0.05%. These values ensure proportionality between dose and impurity risk. Attachment 2 offers practical examples on how to report impurity data. It includes rounding conventions, total daily intake calculations, and actions required when thresholds are exceeded. For instance, an impurity at 0.124% in a 0.8 gram per day dosage translates to 1 milligram per day. This requires identification but not qualification unless safety concerns dictate otherwise. This decision tree guides regulatory decisions. Are impurity levels above identification or qualification thresholds? Is the impurity structure known? Does it pose clinical risk? Depending on the outcome, steps may include conducting toxicity studies or reducing the impurity level to acceptable thresholds. This tree is a vital tool for both regulators and industry during dossier evaluations. To recap, ICH-Q3A R2 provides a harmonized approach to controlling impurities in new chemical drug substances. It ensures clear classification and rigorous assessment of impurities. Validité analyticale technique. Justified specifications and impurity listings. Qualification of safety-impacting impurities. Compliance with this guideline not only fulfills regulatory expectations, but enhances the robustness and safety of pharmaceutical products.